Okay, so we're going to have a look at the answers to these questions. So the first question says, bottles of algae in pH indicator were placed at different distances from a lamp. The pH in each bottle at the start was 8.4. The pH values after an hour are shown in the table. So that's telling us that we have our algae in its bottles, and we've got a lamp, and of course the algae is photosynthesizing. Okay, so let's just remind ourselves of the photosynthesis equation. So we have carbon dioxide plus water using sunlight energy, which is being trapped by the chlorophyll, is being turned into glucose and into oxygen. And so the link here in the question is pH to the carbon dioxide, because when carbon dioxide dissolves in water, we know it becomes slightly acidic. And so let's think about what's happening here. So basically, the closer to the lamp it is, the more photosynthesis is going to be occurring, so the more carbon dioxide is going to be used up and taken out of the water. So we would expect the one closest to the lamp to be becoming the most alkali. So remember that pH 7 is neutral and 8 or more is alkali, and below 7 is acidic. And so what we're looking for is the highest number, which tells us the most alkali. So the most alkali here is B. So we would put for our answer here, the answer is B, because it is the most alkali, so has lost the most CO2. Okay, and we can add there that that has been used up in photosynthesis. Okay, so question 2 here, it says the graph shows how the rate of photosynthesis changes in some pondweed as the light intensity increases. So let's just make sure we understand our graph first. So we've got light intensity increasing along the bottom, the rate increasing on our y-axis, and here we've got two graphs. We've got one which they're showing us has a value of R here, and then that flattens out as Q at 15 degrees. And then we have another line which is P here. Again, that's pretty much a straight part of the line, and then that flattens out at a higher rate, and that is at 25 degrees. Okay, so it says, look at the slope labelled R. What does this line tell you about the relationship between light intensity and the rate of photosynthesis? Okay, so the thing that we have to notice here is that this is a straight line, and because it's a straight line, in maths we write that as linear. Okay, so this is linear, and that tells us as well that these things here are directly proportional. Again, that's a phrase you might use in maths. Okay, and what that means is if light intensity increases, rate of photosynthesis will increase by the same amount. So this could be, for instance, 1. Okay, so if light intensity increases by 1 to 2, we would expect rate to increase by the same amount each time. So now it says define the term limiting factor. Well, we know that the limiting factor is something that stops the rate of photosynthesis increasing. Okay, so remember my barrel analogy. So you've got the sides of the barrel, so the shortest side is always as far as the water level will get to within the barrel. Okay, it doesn't matter whether we make the other ones bigger, it really makes no difference. We can only go up to the shortest length on the barrel, so that's the limiting factor. So what is the limiting factor at point P? Well, we know there are several limiting factors to do with photosynthesis. We have temperature, we have carbon dioxide concentration, we could have how much chlorophyll is present in the plant itself, and we also have light. Now we know that we've used light already, so it has to be one of the other ones. So the most obvious one to use is carbon dioxide concentration. oh no, sorry, at P, no, it's not carbon dioxide concentration. It is, of course, light intensity, because as we increase light intensity, we know that the rate of photosynthesis is increasing. Where we get to this point here, then something else is kicking in. Okay, so it's going to be light. Then it says, why can the factor in C limit photosynthesis? Well, we know that light energy is required for photosynthesis to occur. So if we remove the light energy, then of course photosynthesis is going to stop. Okay, let's have a look at the next question. So what is the limiting factor at point Q on the graph? Now the clue here is these two lines with the temperatures indicated. If they've indicated, they're going to ask you about them at some point. So the difference here between these is obviously temperature. So the limiting factor at point Q is going to be temperature. Okay, so why can factor E, so temperature, be limiting for photosynthesis? Well, of course, photosynthesis needs enzymes. And we know that enzymes work faster as temperature increases. But we also know that that only works up until a certain point. And once we get to a certain point, if it's too hot, until it gets too hot, and then we know that the enzymes denature. Okay, basically they lose their shape, the active site changes shape, and so the substrate no longer fits onto the active site, and at that point then the enzymes are denatured. Okay, so state one other limiting factor of photosynthesis. Well, here is the other one that I mentioned wrongly a moment ago, is carbon dioxide concentration. Okay, right, question 3. In an experiment, equal amounts of pondweed were placed in bottles at different distances from a lamp. So that's the same as that experiment. Let's have a flick back to my quick little diagram. Here it is. Okay, same experiment as we did earlier. The gas that was produced by each piece of pondweed was collected. The table shows the results. The light intensity is in inverse proportion to the square of the distance. The relative light intensity at 30 centimetres is 10. Use the inverse square law to calculate the light intensity at the other distances. So the inverse square law is using this equation, okay, where we know that the light intensity of the new position equals the light intensity of the original position times the distance squared of the original divided by the distance squared of the new. So they're asking us to calculate the light intensities at each one of these distances. Okay, so let's do the first one. So the light intensity of the new, which is what we're trying to work out here, is the light intensity of the original, which we know was 10, times by the distance squared of the original, distance squared was 30, divided by the distance squared of the new. The distance squared of the new is 60. Okay, so if I work out that calculation, that comes out as 10 times 900 divided by 3,600, which gives me 9,000 divided by 3,600, which gives me a value of 2.5. So my light intensity is 2.5. Okay, let's do the next one. Okay, now this one, it doesn't matter whether I take the original here, 10 and 30 as my values, or I could use these ones as well actually, because now this could be my original values and I'm shifting my light further and further away each time. Okay, I'm going to use the original ones, but it really doesn't matter which ones you do. So for that one, it's going to be 10 times 30 squared again, okay, which is my light intensity and my distance, divided by my new distance now is 90. So that's going to give me 9,000 divided by 8,100, which gives me a value of 1.11. 1.11. Okay. My next one, okay, is now going to be 120. So this time, okay, again, I've got 10 times 30 squared, this time divided by 120 squared. So that's going to give me 9,000 divided by 14,400, which gives me 0.625. Okay, I could round that to 0.63 if I wanted. Okay, the next one, 150. So this time I'm going to have 10 times 30 squared divided by 150 squared, which is going to give me 9,000 divided by 22,500, which gives me a value of 0.4. Okay, so I'm going to put that into my table. Okay, so they are my relative light intensities at each of these distances. Okay, so now it says draw a graph to show the relationship between relative light intensity and the rate of photosynthesis. Okay, so you could have done this using Excel on the computer, or you can get yourself some graph paper. So I'm going to get myself some graph paper here. Okay, and it wants me to do relative light intensity and rate of photosynthesis. So I need to work out which goes on which axes. Now I know whenever we draw graphs, remember, the first column is always the independent variable, the thing that I change, and the subsequent columns are always going to be my dependent variables, okay, the things that I'm measuring. Okay, so this is what I'm measuring, and it's asking me to plot relative light intensity against rate. Okay, so I'm going to have to work out my rates first of all. Okay, so let's do that first. Okay, let's work out this first one here. So this is telling me that after 40 minutes I've collected 20 millimeters cubed of gas, and I need to work it out how many millimeters cubes of gas I've collected after 1 hour. So here, let's do the first one. So in 40 minutes I've collected 20 millimeters cubed. So that means that if I work it out for 1 minute, I will have collected 20 divided by 40 millimeters cubed. That's just like you would do for algebra in maths. If I divide this side by 40, I have to divide this side by 40 too. Okay, so now I want 60 minutes because I need an hour. So 60 minutes equals 20 divided by 40 times by 60. And again, I could times each side by 60. So that gives me a value of 30 millimeters cubed per hour. Okay, so that's my first one. Okay, let's do the second one. So this time in 5 minutes I've got in 40 minutes I've got 5 millimeters cubed. Okay, so in 1 minute I've got 5 divided by 40, and in 60 minutes I've got 5 divided by 40 times by 60, which gives me a value of 7.5. Okay, let's put that in my table. Right, my next one, I've got, after 40 minutes I've got 2 millimeters cubed. So 40 mins equals 2 millimeters cubed. So therefore 1 minute is 2 divided by 40, and 60 mins is 2 divided by 40 times by 60, which gives me a value of 3. Okay, next one, after 40 minutes I have 1 millimeter cubed, so I do exactly the same. 40 minutes is 1 millimeter cubed, therefore 1 minute I'd get a 40th of a millimeter cubed, and in 60 minutes I'd get a 40th times 60, which is 1.5. And then finally, in 40 minutes, this time I've collected 0.8 millimeters cubed. So in 1 minute, that is 0.8 divided by 40, and in 60 mins, that's 0.8 divided by 40 times by 60, which gives me a value of 1.2. Okay, so I've worked out my rates. Okay, so now I can move on to do my graph. Okay, So using a ruler and a pencil, okay, I'm going to draw my axes. So there's my axes, my y-axes and my x-axes. Just going to draw lines to start with. Now I need to work out, remember, relative light intensity and the rate. So this time, okay, my graph is actually going to be light intensity, which has moved from my independent variable because it's calculated, to what I've measured, which is my dependent variable, my rate. So the maximum light intensity I've got is 10. Okay, so on my x-axis, because my independent variable always has to go on my x-axis, I need to go up to 10. So I'm going to start with 0 here, and let's see if I could do it in ones. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so that works. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And I mustn't forget to label it, because you lose marks if you don't have your labels on your axes. Okay, so there's my relative light intensity, and now my rate. Okay, let's look at my figures. So my maximum figure is 30, so I need to go up to 30 on my y-axis. Okay, so could I go in ones? No. Could I go in twos? 2, 4, 6, 8, 10, 12, 14, 16, 18, 22, 24. Yeah, I could just make that a little bit longer. So let's do that in 2. So 0, 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24, 26, 28, 30. Okay, and again I'm going to label this. Okay, so it is rate of photosynthesis, and they have given me units, so I must include my units. If I don't include units, I will lose marks. Okay, so there's my axes all drawn out, ready to go. So now I'm going to plot my points. So the first one, okay, let's have a look. So I've got 10 and 30. So my light intensity is 10, my rate of photosynthesis is 30. So if I go up to there, I'm just going to put a cross, and I have to be quite accurate when I plot these in exams. The next one, 2.5, 7.5. So here's 2.5. And where's my rate of 7.5? So it is, there's 7. So 7.5 is going to be there. Okay, so 2.5, let's go up. 7.5 there. Okay, my next one, light intensity of 1 and rate of 3. So 1.1 is going to be there, 3 is there. Next one, 0.6, so that's 0.6 is going to be there, 0.625, and a value of 1.5 is going to be there. My next one is 0.4, so 0.2, 0.4, a value of 1.2, which is going to be there. Okay, so there are my points plotted. So now I'm going to draw on my line. Now I could use a best fit, okay, which is where I have the best fit line which passes through the majority of points, or it looks okay actually at this point here that I can just join them up with my ruler. So there we go. So there is my line. Okay. Right, so part C, it says, how does your graph show that the rate of photosynthesis is directly proportional to light intensity? So as we talked about earlier, directly proportional means that it's a straight line, and that it's linear, okay, and also it shows that it passes through my origin, okay, which is my 0, 0. So if there's no light, there's no rate, which is what we'd expect. Okay, so straight line, so it's linear and it passes through the origin. Okay, Right, so final question, question 4. So a bulb gives out a light intensity of 4,000 lux at a distance of 5 centimetres. At what distance must the bulb be to produce a light intensity of 750 lux? Give your answer to the nearest half centimetre. Okay, so I need to use my equation again. Okay, so let's look at that equation. So my new light intensity equaled my original light intensity times by my original distance squared divided by my new distance squared. So let's put in the numbers that they've told us. So they're telling us that it's the original distance, it gives out 4,000 lux. Lux is just the measurement of light intensity, and the distance was 5 centimetres. So let's put those in. So 4,000 times by my original distance, which was 5. Remember, I need to square that in my answer. Okay. And they're also telling me that the new light intensity is going to be 750 lux. So the thing that I'm trying to calculate here is my new distance. Okay, so I just need to rearrange this equation and then solve it. So you can imagine that this is x. If you wanted, you could replace it with an x. So what am I going to do to solve this? I have to times each side by d squared nu, or I could call it x if I wanted. So I'm going to have 750 times d squared nu equals 4,000 times 5 squared. Okay, so that means that now I want to get rid of the 750 from this side. So I divide each side by 750. So now I've got d squared nu equals 4,000 times 5 squared divided by 750. So that equals 100,000 divided by 750. So that tells me that d squared nu equals 133.3. But I don't want d squared, I want d. Okay, so what I have to do therefore to calculate d, so the new distance, is therefore the square root of 133.3, which gives me a value of 11.5. And it wanted it to the nearest half centimetre. Okay, so there we go, 11.5 is my answer.